I want to welcome everyone to today's OCB webinar. My name is Heather Benway, and I manage the OCB Project Office, which is hosted by the Woods Hole Oceanographic Institution. The project office includes two additional staff members, May Mahegan, who manages OCB communications, and Mary Wysowski, who manages administrative and logistical operations. I'd also like to acknowledge the sustained support of NSF chemical and biological oceanography programs, as well as the NASA Ocean Biology and Biogeochemistry program, which make the OCB program possible. We've been in operation since 2006, so that's a long time to support this program, and this community's done a lot of great things since then. Today, we are also joined by OCB SSC member Claire Reimers from Oregon State University. She's a member of OCB SSC, and she's serving as my webinar co-host. To give you a sense of the scientific breadth of OCB, these green icons depict our overarching science areas. And you can find more specific research questions of community interest on our website. And I definitely encourage you to visit the website. There's a lot of information there. OCB webinars typically have topical themes with 2 speakers per webinar. And to broaden access, we record all of these webinars and post them on our YouTube channel. We share information about our webinars via our website, our e-newsletter, and Twitter. We'll have 2 more webinars in 2020. On November 17th, we'll have an update on the status of the exports field campaign and brief updates from PIs of NASA and NSF-funded exports projects. Then in early December, we'll have 1 more webinar. It's a secret right now. We're currently finalizing decisions based on recently submitted abstracts, and we'll share the details on that as soon as they're available. Then we're going to pick back up in January of 2021. So please do continue to submit applications. May can post the link to submit abstracts right here in the chat. And you can also find that on our website. Oops. We invite you to engage with the OCB network in lots of ways. They're all listed here. Our website's probably the most comprehensive source of information and history of the program, but I encourage you to sign up for our biweekly e-newsletter, which is a great compilation of information on science updates and community news, science planning, jobs and postdocs, funding opportunities. And also follow us on social media. Our Twitter handle is listed here, as well as our YouTube channel, which is where these webinar recordings will be posted. You can always also reach us by email, which is listed here as well. Just a little quick primer on how to use WebEx. This is the screen that you should be seeing. Please note that all attendees are muted and video is off, just to reduce background noise and distraction. You cannot unmute yourselves. You can only be unmuted by the host. If you can see the phone and dial-in information. I think May has posted that in the chat also, but it's also under this dot here where it says audio options if you're having a hard time with the video. The X is obviously how you leave the webinar. And you can view. If you click on participants, you can view all the panelists. You will not be able to see the full list of attendees, though. I can tell you that we had between 160 and 170 people who registered for the webinar, so I think we have really excellent showing today. On the bottom right-hand side here is probably the most important thing for you to note. This is the chat with this little message icon. You will use that to ask questions, but please be mindful of who the chat recipient is of your question. For example, if you select host, the question will only come to me. If you select all panelists, it will go to speakers and our SSC chair and myself, so all of us as panelists. And if you select all participants, which is also perfectly fine, it will go to everyone on today's webinar. We will be moderating the questions today, and if we don't get to all of your questions for the speaker, we'll be sure to share them with the speaker so they can follow up with the attendees after the webinar. And we'll also be using the chat to post references and other helpful information throughout the webinar, so I definitely recommend that you keep it open. So the theme of today's webinar is productivity and marine snow dynamics. Today's speakers are Amanda Timmerman from University of California, San Diego, and Klas Ove Möller from Institute of Coastal Research at the Helmholtz Center for Materials and Coastal Research. I'm going to have Claire introduce our first speaker, Amanda.
Hi, attendees. I'm Claire Reimers, and I'm very happy to be here to introduce our speakers. So Amanda Timmerman is currently working, as you heard, at UCSD as a research associate at Scripps Institution of Oceanography. She earned her BS and MS at the University of Hawaii and her PhD at the University of Victoria. Her research focuses on phytoplankton's role in biogeochemical cycling and what factors drive phytoplankton variability. She likes to take an interdisciplinary approach connecting physical oceanography to phytoplankton production. She combines shipboard sampling, satellites, and model output to help her answer research questions. And she'd like to continue that kind of research and looking at the importance of mesoscale dynamics and the role zooplankton play on controlling phytoplankton distributions. She's participated in cruises in 4 oceans, but she says she's missing the Indian Ocean and hopes to get there one day. She's also likes to spend time outside hiking, backpacking, sailing, and paddleboarding. And she reports since the global pandemic started, she's been busy in the kitchen, taking up baking, and her favorite creation so far is sourdough cinnamon buns. I think I can almost smell them. So please welcome Amanda for our first talk today.
I just want to say thank you for having me. This is quite the honor, and I'm excited to talk about my research that I've been working on recently. So I'm interested in the biological carbon pump, where in the surface waters, phytoplankton photosynthesize, taking up carbon dioxide to grow. A fraction of that carbon leaves the surface water, taking it out of contact with the atmosphere. This can help regulate climate and have insights into possible climate change scenarios. I'm interested in the Northeast Pacific. So on the right is Seattle, and I participated in the Line P time series cruises. They go out 3 times a year. And I was focused on the June and August cruises, starting August 2014, and I have data until August 2017. This Line P time series has 5 major stations. P4 is the coastal station, P12, 16, 20, and 26. I'll be talking about these stations throughout the seminar. So the lower numbers are on the right-hand side and closer to shore, and then the offshore ones are on the left, and that ends with P26. The color in this is bathymetry, so they're all quite deep. So I'm really interested is understanding how variable chlorophyll is in the region and what causes the spatial and temporal variability in phytoplankton biomass and productivity. As it turns out, I started sampling during a unusually warm temperature anomaly that started forming in the winter of 2014. It's been named the blob. So this is a figure for sea surface temperature anomalies in the region in February 2014. So you can see that the temperature anomalies are up to 3 degrees, and the gray line is where the Line P transect is located. The dominant currents in the region is the North Pacific current that comes from the west to the east. When it hits the coast, it bifurcates. And so the northern part forms the Alaska gyre and the southern part forms the California current. There are gradients along this transect. So on the coastal P4 station, macronutrients are drawn down by phytoplankton. However, iron concentrations remain high. And it's also influenced by coastal upwelling. In contrast, on the offshore station in P26, macronutrients remain high throughout the year and iron concentrations are low. For this talk, I'm mainly interested in the offshore region. So I will only be showing P12 to P26 for the rest of the talk. People have been interested in iron concentrations in this region. This is a figure from a paper in 2006, where they added iron and then followed how phytoplankton responded to that over 20 days. So the x-axis is days since the iron was added, and then the y-axis is chlorophyll concentrations, and they looked at 3 different size classes. So if we look at the 2 smaller size classes, which are triangles and diamonds, you can see after the enrichment, those that biomass, that chlorophyll concentration remains quite low throughout the entire time period. And the larger phytoplankton, which are squares, increased over that iron enrichment period. So that shows that iron is limiting these larger phytoplankton, and when you add iron, that there's a shift in community structure from smaller phytoplankton to these larger phytoplankton. And it was also shown that zooplankton grazing wasn't sufficient to prevent this phytoplankton bloom. So if you look at the more normal, not iron additions, this is surface chlorophyll for the top 20 meters. For again, the x-axis is the station, so we have P12, the more coastal side on the right, to P26 on the offshore on the left. And this is, again, surface chlorophyll for 3 different seasons, which are the colors. So winter, spring, summer for when the cruises go out. And this covers 46 years from 1988 to 2005. So you can see there's not really much variability in terms of seasonality or interannual variability. If we look instead at higher resolution, so in the 50s and 60s, there was a weather ship located at P26, so it allowed for higher resolution temporal, higher resolution measurements. So on the x-axis, we have years from 1965 to 1976, and the y-axis is chlorophyll, and you can see that most of the chlorophyll concentrations are low, between like 0.3 to 0.4, with some periods of time where chlorophyll does increase and is higher than 1. To compare that to what has happened more recently, so from August 2014 to 2017, this is, again, on the x-axis is stations. So we have P12 on the far right to P26 on the far left. The y-axis is surface chlorophyll. The different, so the major stations, P12, 16, 20, and 26 are slightly larger symbols, and then the different colors represent the different cruises. So generally the darker color represents the June cruise, and then the darker one is the August cruise. So you can see there's quite a lot of variability through this 3-year period. And in fact, if we look at just P26, the average for this time period is 0.65 milligrams per meter cubed, and the standard deviation is nearly as large as the mean. Putting that into context with the historical data, so from 1959 to 1977, the average is 0.43, which historical papers have talked about, but they really haven't talked about the variability, which is, again, nearly as high as the mean. So I'm only focusing on 3 different cruises, only focusing on June cruises for this talk. So let's go through those. So this is June 2015, where chlorophyll is increasing as we're moving offshore. This is June 2016, where chlorophyll is much higher and more variable. And in fact, the highest chlorophyll concentration was at P26. And then the last year is June 2017, where chlorophyll is low and invariant throughout the entire transect. So these are only like single cruises, only 2 times a year. So to increase the spatial coverage, I turned to satellites. So this is daily chlorophyll for June 2015 from a merged chlorophyll satellite product. So you can see in between all the clouds, which is the white, that there is areas where there's higher chlorophyll. So maybe doing one transect twice a year is enough to really capture the amount of variability that's occurring offshore in terms of chlorophyll. You can see it's quite cloudy out there during June. So instead of taking daily, I instead took 8-day averages of chlorophyll for around the time period that I was sampling. The top plot is June 2015, the bottom plot is June 2016, and the color is satellite chlorophyll. So you can see that chlorophyll is quite variable throughout the entire region, and we actually see that there are cohesive features, for example, the one where I have the arrow pointing at. If we look at June 2016, it follows what the shipboard data shows, in that chlorophyll was high and variable throughout the region. So if we take a closer look at June 2016, the x-axis is station again, so P12 is more closer to the coast versus P26 offshore, and this is surface chlorophyll for those 4 major stations. I also conducted incubations. So this one is 13C incubations. That is a measure of primary production, and it generally follows the same trend as chlorophyll. There's also, I did 2 methods that estimate carbon export. So I did oxygen to argon ratios as well as 15 nitrate incubations, and those as well follow the same trend as chlorophyll. And then the last one is 18O incubations, which is a measure of gross primary production, and those also follow the chlorophyll trends. So when you have higher chlorophyll, you also have increased productivity and carbon export. So in fact, there are many parameters that are not constant offshore, and there's more variability. So the question is whether there were iron inputs that caused this variability in chlorophyll. I can't say directly, however, I can use indirect evidence, and so I'm only using one for this presentation. And it's looking at phytoplankton counts and samples where they are grouped into 4 different classes. So we have diatoms, dinoflagellates, flagellates, and then other. The top plot is chlorophyll, and then the bottom plot is the relative abundance of those phytoplankton species. So the color I want you to look at most is the blue one, which is diatoms, which are generally larger phytoplankton. And at P26 and P16, the diatom relative abundance is around 30%, whereas typically the relative abundance is less than 10. If you remember back in my slide about iron additions, that you'll have a change in species composition to larger phytoplankton when iron is added. So this implies that there were iron additions at least at P26 and P16. So there was likely iron, and so where did it come from? And it can come from one of 3 ways, I think. The first being from above. There are a couple papers, this Boyd et al. In 1998 showed that probably Asian dust or dust from Alaska helped control some of the variability that they saw in phytoplankton blooms. And then Roberta showed that volcanic ash stimulated a phytoplankton bloom. However, I looked at UV absorbing aerosols, aerosol optical depth, and model to look at dust, and I didn't see any correlation between dust and the variability that I saw in this region during those 3 years. Another way that iron can come into the area is from horizontal. So I looked at eddies by examining sea surface height anomalies. And so sea surface height anomalies is the top plot in color, and then the bottom plot is satellite chlorophyll. This is June 2015. So we can look at this by looking at where there is a high in sea surface height anomaly, and it doesn't really correspond to anything in particular in terms of chlorophyll. If we look instead at the high chlorophyll and see how that corresponds to sea surface height anomalies, again, I don't really see any correlation between the two. If we look at June 2016, I've highlighted in the black box the sea surface height anomaly, low sea surface height anomaly, which corresponds to a hole in chlorophyll. I thought this was pretty cool when I started going through how well they correspond, and this isn't something new, but I thought it was interesting. So sea surface height anomalies can explain some variability. For example, this. However, there is a paper by Shelton et al. 2011. This is showing the eddy tracks for eddies that have lifetimes greater than or equal to 16 weeks from the period of 1992 to 2010, where the black dot in the middle is where P26 is located. And you can see this region has an eddy dead zone where eddies don't normally go. So although eddies can explain these localized trends in chlorophyll, they really can't explain the overarching trends that we see in chlorophyll throughout the entire region. So the last place that it could come from is below, which I think is the most likely. This is a TS plot, which I'll show quite a few of these. So on the y-axis is temperature, and the x-axis is salinity, and then in color is chlorophyll. This is all underway data from June 2016 cruise, where you can see the higher chlorophyll is located where the temperature is lower and the salinity is higher. To increase the spatial coverage, I turned to models and satellite. So this is another TS plot. So temperature on the y-axis and salinity on the x-axis. This is for June 2015. The temperature and salinity came from a model, and then the chlorophyll is from satellites. And you can see that the higher chlorophyll is actually located in a range of temperatures from around 10 to 13 degrees. And really you have two water masses. So maybe you have a higher temperature and maybe a lower salinity, and then a lower temperature and higher salinity. So if we look at the other 2 years, June 2016 had a similar pattern as June 2015, where you have that higher chlorophyll water at that certain temperature range. And then in contrast, we have June 2017, where the temperature was low and the salinity also was higher, and you didn't get those mixture of water masses between the two, as well as not having higher chlorophyll. So what mechanism could be responsible for this higher chlorophyll regions that we see? So this is sea surface temperature again, and the arrows are geostrophic currents. And so I just wanted to point out this one feature in the black box. So what I'm going to do is go from sea surface temperature and then fade into the satellite chlorophyll. So look in the black box. And they align really, really well. And I'm going to go back to sea surface temperature so you can see the transition again. So chlorophyll is really located at these temperature gradients. And so our idea, is that fronts could be the possible mechanism, which probably isn't a surprise based on my title. So what we're looking at here is June 2015 and June 2016. So June 2015 is the top figure and June 2016 is the bottom figure, where temperature is in color. And then what I've done is all of the black dots are locations where there's higher chlorophyll, greater than 0.6. And then all the white dots are the locations where fronts are located based on a criteria of 1 degree change in temperature over a 3rd degree distance. So I just want to point out a few of them where there are fronts and there's also chlorophyll near. You can see that the chlorophyll is generally in the colder water. So So this is 2 years. And then this is the last year, June 2017, where the top figure, the color is chlorophyll, and the bottom figure is similar to the other ones where the black dots represent higher chlorophyll and the white dots represent front locations. And if you'll notice on the bottom figure, there are no front locations in the region at all during this year. And that also corresponds to having low invariant chlorophyll in the region. So I think this helps point to that maybe fronts are a possible mechanism for this region. This is a new idea for the offshore area. They've shown like coastal regions and maybe even transition areas between like the coastal waters and the offshore waters, but it hasn't been put forth for this high nutrient, low chlorophyll area. So I'm interested to hear what other people think on this idea. And I guess we have two theories on, well, two as of right now. I'm not sure, there's probably more, but two thoughts on what could be causing this. One could be the location of the North Pacific Current. So this is sea surface temperature from 2015 at the top, the middle is 2016, and the bottom one is 2017. They're all the same scale. So maybe we're seeing a shift in the North Pacific Current from more northerly to more southerly is one idea. Another idea is that this is maybe dominated by the blob, so that warm temperature anomaly. So this is 2015, 2016, and 2017. Again, plotting sea surface temperature anomalies, where you can see that 2015 is completely dominated by the blob, and then it decreases as you move through time to the present. So maybe this is an analog for what the future oceans will look like with the warmer temperatures. Maybe we'll have more fronts. Who knows? That's just a theory. So like I said, I'm interested to hear what other people think about this. So in summary, chlorophyll is more variable offshore than the historical view. This higher chlorophyll is associated with gradients in temperature. I have indirect evidence that there are iron inputs, and none of the other methods can really explain the variability. So I think it's pretty likely that fronts are the source for this iron. So things that can be done in the future, there are so many, but understanding what causes these fronts to form, which I have a couple ideas. Not all of those fronts were associated with higher chlorophyll. So looking at what makes a front biologically important. How important is zooplankton grazing? I didn't really look at that too much. It'd be nice to have high resolution iron measurements at the base of the mix layer or at the base of the photic zone, and then really to get estimates of vertical mixing to understand at these fronts, what is the vertical motion of water? How deep could this water be coming from? This iron concentrations are quite low, need to be quite low to support the variability in chlorophyll that I'm looking at. So I just want to say thank you to Roberta, my co-author on the paper that we just submitted on this, and a bunch of other people. And I just want to say thank you.
Great, thank you, Amanda. So now can enter your questions in the chat. I can relay them to Amanda. And maybe while you're thinking, I'll ask a question. In the historical record, you showed us chlorophyll, but are there other times in the last 50 years where there have been these unusual warm spells, these temperature anomalies that, like the blob, or is that really a new phenomenon?
I think they have had some in the past, probably not within the recent past, but there definitely have been times where there's been higher temperature water in the region. This particular one was fueled by having a high pressure system located in the region over winter, so it caused less mixing, and so there was less warm water being removed from the area. So
great. Well, I'm not seeing any questions coming in my box. Is that a problem with my viewing? Or?
No, people are sending them to the host. Hold on a second. I'm starting to get a bunch here. Okay, from Jen Fehrenbacher, if your findings are linked to marine heat waves like the blob, do you find similar patterns in 2019?
Yeah, so I only looked at June 2015 to 2017, and I have not looked at more recent years or even in past years. It's a little bit unfortunate that I started in 2014, but that's definitely something that can be looked at more. I know there's been more weird things going on in the Pacific after I stopped sampling.
I have another question about iron sources from Ken Bissler. What about lateral transport of suspended particulate iron associated with margin particles? Lamb and Bishop talked about this in the late 2000s.
Yeah, transported. You know, I didn't really see anything. I don't know if you have maybe better ideas on how to quantify this or look at this more, but I'm not sure.
From Matthew Rao, I'm curious about what the sources of the iron is at these fronts. You know that chlorophyll correlates well with the temperature fronts, but why is iron higher at these fronts?
So I think there's vertical motions associated with these fronts. I don't think that the iron is coming from the ferrocline. That's way too deep. So I think my theory, which I can't prove because we need higher resolution measurements of iron, but I think there's enough iron just below, closer to the surface, that could be brought up to supply iron for these. I mean, the variability in chlorophyll is not like high like what we get at the coastal regions, so you don't need a ton of iron to be able to do this, to fuel this variability in chlorophyll. So maybe it's coming from, I'm not sure, maybe it's coming from, as phytoplankton degrade as they are sinking, it's accumulating small amounts of iron underneath the mixed layer and photo depth below. It's hard to say. I'm not sure. This is just an idea I'm putting forth, and so I'm interested in hearing what other people will think about this new idea for the region.
Well, your idea has spawned a lot of conversation here in the chat, and I think we're going to have. I'm gonna have to send you all of these questions. I have several more that I'd love for us to get to, but we need to move on to Klas's talk now in the interest of time. But I will be sure to make sure that Amanda gets all these questions and can respond to you.
Great, thanks so much.
So I am going to have Claire introduce Klas while I give him the presenter baton here.
Okay. Well, I'm happy to introduce Klas. He is currently working at the Institute of Coastal Research at the Helmholtz Center, Geesthock in Germany. He earned his PhD in biological oceanography at the University of Hamburg using underwater imaging systems to resolve small-scale distribution patterns, trophodynamic interactions, and the impact of climate change on zooplankton. He's worked as a guest scientist at the Woods Hole Oceanographic Institution, the University Center in Svalbard, and the Scripps Institution of Oceanography. At the present, he is a young investigator group leader and focusing on various mechanisms influencing the efficiency of the biological carbon pump and the productivity and biophysical dynamics of marine ecosystems. His expertise is spatial and temporal high-resolution observations, including towed and stationary imaging systems, which he's going to merge with remote sensing approaches in the future. He reports in his free time he is into long-distance running and spends as much time as possible on, at, or under the sea. And the global pandemic has kind of forced him to keep dry at home, but he reports during this time, he's tried to deepen his knowledge in artificial intelligence. However, he still loses 9 out of 10 matches against the Czech computer. So with that in mind, we now welcome Klas.
Thanks a lot. And first of all, thanks the whole OCB committee for having me and all of you for attending Amanda's interesting talk and mine now. and I'm going to talk about the active controls of zooplankton on marine snow dynamics. So first of all, why do we care about sinking aggregates? It's basically known that estimates of carbon flux in the oceans are essential for our understanding of global carbon budgets. And furthermore, these marine aggregates of biogenic origin, known also as marine snow, are considered to be the major vector of vertical carbon transport within the framework of the biological carbon pump. And so due to this, it's necessary to elucidate the different mechanisms driving the particle transport, including passive and physical mixing and also sinking of particles, but also the active and biological mechanisms like zooplankton feeding directly on particles, degradation of particles, but also vertical migration. And both together here can explain the question mark, which is still a very bold question mark due to the limitation of traditional sampling methods and their limitation in the spatial and temporal resolution. And furthermore, which is also quite interesting and necessary to study, is not only the vertical mixing, but also the advection. So since coasts are high productivity areas, I'm also really interested in how much of these particles are driving or fueling the deep ocean carbon pump. So the overall research goal here is to develop a predictive understanding of how biology, chemistry, and physics together impact the efficiency of the biological carbon pump in the present, but also in the future ocean under the framework of a changing marine ecosystem. And to do so, we need to perform quantitative measurements of the controlling mechanisms within the biological carbon pump. So really the quantitative thing is really important here, and therefore we also need to combine ecological, biogeochemical, and also physical field observations with remote sensing, but also modeling approaches. And due to the recent developments in technology, we have come to a state where we have now acoustics and also imaging systems. So we really have the tools available and in our toolbox to improve the quantification of the carbon pump. As a first step, what we did was some while ago to attach imaging systems to high-speed tow bodies, like you can see in the left panel here on the Triaxis, which we towed along a star-shaped transect in the Bornholm Basin in the Baltic Sea to the right here. And what we found was a very high concentration, thin band of particles aggregated directly at the density gradient at 60 meters. So the particles really aggregated and settled at this density gradient. And what we also found in this distinct area was copepods and a high concentration of copepods being directly attached to marine snow particles and showing a feeding behavior. So their antenna spread to the side means they are somehow interacting, there is a potential feeding. And as Morten Iversen also showed in the lab to the right is that they are actively feeding on particles and are kind of really happy when they find something to feed on. So there is an indication that zooplankton and especially copepods are feeding on marine snow. And as a next step, we went to the North Atlantic Ocean and did a cruise across several basins, and were able to separate snow particles, which are traditionally grouped into one large category into separate categories, including regular snow, so these yellowish fluffy in the upper left, also krill fecal strings in the upper right, and also marine snow coming from Phaeocystis bloom origin to the left, and also from diatom blooms, so marine snow of diatom origin, as well as appendicularian houses. And all these categories have shown very distinct vertical patterns. And as you can see on the right side here, I show the very distinct difference between diatom snow and Phaeocystis snow. And the plot gives you the vertical depth on the y-axis over time. So you can see two toyos with this imaging system. And we find very high concentration of both snow types. And if we now look at the vertical patterns, there's a clear separation with diatom snow found mainly below 400-meter depth and Phaeocystis snow above. So this might also indicate some kind of feeding at the surface by zooplankton on the diatom snow. And exactly, we again found particles colonized by copepods. And on the left side, in this large image, you can see Onkea, which is a cyclopoid copepod, which was found exclusively on marine snow of Phaeocystis origin. And on the right side, this orange guy is Microcitella, a copepod which is known to colonize particles, and that was found in the upper water layer and exclusively only on marine snow of diatom origins. So there's also a clear niche separation. So it's nice to see copepods showing a potential feeding interaction with particles, but we still don't know how much is the impact. And to find this out, we teamed up with Camilla Svensson from the Tromsø University and some other collaborators, and we went in 2 years into a northern Norwegian fjord, the Bals Fjord, and we wanted to study here the important or the impact of the tiny copepod Microcitella, which is really in all coastal areas highly abundant, as a gatekeeper for, or if this copepod is a gatekeeper for the biological carbon pump. And so what we did was also doing some kind of field and lab measurement combination. We sampled with nets and water bottles and also a video plankton recorder, so an imaging system, aggregates and copepods in this fjord. We deployed short-term sediment traps and gel traps to measure the vertical flux of carbon and particles. And we did also experiments measuring rates and feeding behavior, as well as reproduction and respiration. And in this talk, I will only focus here on the feeding experiments. So what we found was that there's a strong reduction in the vertical flux, what we measured from the sediment traps, with a strong carbon reduction in the upper 50 meters of 220 microgram carbon per square meter per day. And additionally, we found a very high number of Microcitella copepods in the upper 50 meters, so matching directly with the depth of the strongest reduction. And this concentration was really high, with 1.5 individuals. And then we estimated the grazing impact, and we could attribute 0.03 microgram carbon per individual per day, which can be attributed again to potential grazing impact, and 45 microgram carbon per square meter per day. So the reduction only of this one copepod species of Microcitella can be attributed to the carbon reduction, or 20% of the total carbon reduction in the upper 50 meter within this fjord. So, and what we also found were, again, images and Microcitella being attached in the upper 50 meters on these aggregates, and showing again this feeding behavior. And if we are now looking at the distribution of these copepods in 2017, which was the first visit in this fjord, we see here the depth distribution of individual copepods for each of the image deployments or imaging system deployments. And we see a very stable distribution pattern, so very high concentrations close to the surface. And the same was true for 2018. Here we performed more measurements with a video plankton recorder to obtain a whole daily cycle. But this is really a very consistent pattern with high concentrations at the surface. When we now look at the distribution of particles and krill fecal pellets, we see that during the first year we had a very strong concentration of particles of marine snow aggregates at the upper 50 to 40 meter close to the surface, and nearly no krill fecal pellets. And this picture changed completely in 2018, where we didn't find any marine snow particles. So, which might be due to the fact that we missed the phytoplankton spring bloom, although we went there during the same time. But we found a very high concentration of krill fecal pellets. And the krill fecal pellets also are indicating some kind of daily vertical migration. And if you can see my mouse pointer here, you can see over the course of the day that there's an increase of particles close to the surface and then moving downwards again. So what is Microcitella doing when there are no marine snow aggregates? Clearly, they jump on these fecal string and are feeding and colonizing heavily on these particles. And this is something we are currently also estimating. So the impact of these copepods on krill fecal pellets. But it's not only copepods who we found are feeding on marine snow. And this is really a cool guy. This is Noctiluca scintillans. It's a heterotrophic dinoflagellate, and it's feeding across a wide size range. So it's including not only fecal pellets, but also copepod eggs and copepods itself in some areas. And recently it has been found to perform large blooms due to its high tolerance to anoxic layers and also high temperatures. So it's spreading globally currently. And it's also known to perform these ocean glowing blooms. So these due to its bioluminescence. And recently they have been also found to perform winter blooms in the East Chinese Sea. And so we also know that they are performing quite heavy blooms in the North Sea area. And what you can see on the right satellite image is a very typical sign of a Noctiluca bloom with these finger-like extensions from the coast into the open ocean. And if you're zooming in, you can see that they have a very distinct color. So they are very yellowish during daytime, and they have these very pronounced slicks at the surface. And if we are now zooming in, we can see that there are strong bands of these distributions on a scale of 10 meters. So, and they are extending far on a horizontal plane. And then if you're looking in between these large bands, you can see smaller bands and on a meter scale. So, and even closer looking, then you can see that there are also bands in between these smaller bands on centimeter scale. And this is really something which is currently unknown to now. So how these patterns are driven from large to small scale, if this is some kind of mixture of physics or behavior or purely behaviorally, because this organism is really causing some kind of miracles currently, to me at least, because it also can perform different feeding modes, raptorial feeding, as you can see on the left, single cells, which are engulfing whole strings of diatom chains or fecal strings, and also particles of marine snow aggregates. And on the right side, you can see that they are sometimes also aggregating together, and then they are building huge feeding webs, which are sinking through the water column. And then we did some feeding experiments, how much the impact of this Noctiluca is on copepod fecal pellets. And as you can see, they are heavily feeding here. And you can see the fecal pellets inside their cells. And we also found some kind of very strong functional response supporting this theory that they are kind of a gatekeeper in the ocean, because they are also heavily feeding on marine snow, which is here given on an image by Rainer Kiko, which was done during a cruise in the Southern Atlantic Ocean. So to conclude the first part here, marine snow is a very underrated and maybe also undersampled important food source for pelagic zoo various marine ecosystems, and it might have a huge impact on the productivity of marine ecosystems thereby. And Microcitella, this tiny copepod, has a very strong impact on the biological carbon pump in coastal areas. And furthermore, Noctiluca is a potential gatekeeper of the biological carbon pump in coastal oceans, ranging from temperate to tropical regions, due to the fact that they are currently increasing globally, and there should be really some work be doing. What we are doing in the future, or currently trying to do, is to expand our high resolution imaging to get a better, or even a more better idea of what is driving the productivity, or what is marine snow as a food source for copepods in the coastal ocean. And so we attach imaging systems to landers to do stationary sampling and high temporal resolution, but also to autonomous systems like the wire walker. And what we are getting from this system is a bunch of many images. We're getting almost thousands of images in an hour in coastal areas. And so all these images are sometimes quite nice, but you don't want to really, or you really don't want to look at all these images. And so we are also digging into artificial intelligence and machine learning algorithms for real-time classification. And what we are doing is trying to group these these individual regions of interest on each frame into different categories you can see on the left here. And this is hopefully working quite well in the near future. And then if we get this going, then we can make some kind of high resolution time series to get an idea about the biodiversity in coastal ecosystems and also of invasive species, which might be introduced into certain areas, and but also get some kind of idea of climate change effects and poleward shifts, or if some species are getting less abundant in certain areas, and also of the productivity, and if marine snow is really a very important food source in the food web of coastal areas. And recently, Saskia Rühl joined my little team, and she's bringing a new expertise into the field by combining our pelagic observations with her benthic dynamics. And this is really cool because we now really can also look at the benthos, and especially in coastal high turbid areas, we get some idea of resuspension versus deposition of particles. So how much of the flux is really buried in the sediment. And also on the right here, you can see some kind of measurements of benthos organism impacts on the sediment, and this might also give us some kind of idea if certain areas are sinks or sources of carbon in the ocean. So with this, I would like to say thank you and acknowledge some of my collaborators and colleagues like Burkhard Baschek, but especially also Morten Iversen from Marum Avi, Saskia Rühl, Scott Gallagher, George Jackson, who is also really interested in Noctiluca, hopefully still, Camilla Svensson from Tromsø, and many others. And thank you all for listening. And if you have questions, please let me know.
Thank you.
I think I'm going to go ahead and do the questions.
I've seen a couple come in just to me and then a couple come in to everybody. So I'm going to start with Stephanie O'Daly. How do you distinguish between images of Phaeocystis snow and diatom snow?
So it's on many images, it's very easy. Sometimes it is not, but the diatom snow is more shiny. So it's really by color, because the diatom snow, or at least in that area where we sampled, has a lot of these polysaccharides, which are reflected in the images quite well. And so by looking at the images manually, we could distinguish.
From Cordelia Roberts, could the explanation for the high density of Phaeocystis at the surface and diatom snow below 400 meters be as a function of their settling velocity? For example, Phaeocystis suspended particles and diatom snow sinking.
That is a good comment, and that is something we are currently trying to do, because we luckily also have some information from marine snow catchers from that cruise. And this might be also an important source for the different vertical patterns, or for the explanation. Yes, absolutely.
And we have a question from Alexander Bogdansky. How persistent are diatom and Phaeocystis layers in time?
In time, during this cruise, it was part of a deep convection cruise, so the name, and we were looking at this start of the phytoplankton bloom. And so we visited each of the stations over the course of the phytoplankton spring bloom several times. So each of these stations was visited once a week. And so the week after sampling the first time, the layers were disappearing. So it was less than a week. More we cannot say.
Okay, we have another question from Cordelia. Great talk, class. I never knew Noctiluca had a role in eating marine snow. Do you have any idea whether Noctiluca ends up as marine snow, and so ultimately its gatekeeping carbon could at some point add to the carbon flux?
Yeah, this is also really interesting, because this would I mean, it's known for jellyfish and these jelly falls. At some point, they when they die, they sink to the benthos. And of course, I mean, these huge blooms, when they disappear, they have to sink to the bottom. And hopefully, we can get some more information. I mean, we don't have a very good data set to hand right now, but we are really trying to look into this during the next summertime. Hopefully, if Corona is not preventing us from that. But this would be really a big thing. Yes.
We have a question from Karen Wishner about another copepod often associated with particles is on Saya. I'm not sure I'm pronouncing that correctly. Have you seen them on your particles in snow?
Yes, in the North Atlantic Ocean, and we are currently digging into this data set, and what we found was that microcitella was only found on diatom marine snow, and on Thea, or on Kea, I don't know even how to pronounce, is exclusively found on particles of Phaeocystis origin and in deeper areas. So yes, we found this copepod species being attached, and I think there are also some manuscripts describing Onsea on marine snow particles from a Japanese team.
How do you see linking marine snow abundance and size to carbon flux? I.e., can you comment on sinking rate versus size and people like Iverson who don't see a relationship? This is a question from Ken Bissler.
I mean, there are people arguing currently if size is really a big thing. And I think size is important if you know the type of particles. And I mean, from these images I have shown, and the imaging system I have used, I'm not really trusting the size information or the ESD measurements, which is always critical, even if you want to attribute carbon content to 2D images of a 3D environment. But we are also deploying now holographic imaging systems to get a better idea of size. But I think size is important if you're looking at the depth distribution, and that is something we have to do in the future more with these images.
Great. I think that's about all we have time for today. I'm going to send you any follow-up questions that I find here. They're still coming in. But I want to thank you both for excellent talks. And I want to thank all of the attendees who joined us today, and my co-host Claire and my co-host May. Thank you everybody for joining us. Just a reminder, November 17th will be our next webinar. It will be on exports, and that will be, the information on that will be on the website. So please check it out and join us. And we have one more coming up in December before the end of the year. And we are thinking about adding a little chat session after these webinars in the future, just so that we have time to get to more of these questions. Maybe we could really hone in on early career participants being able to have an informal chat with the speakers after the webinars. So we are hoping to implement this in the next couple of webinars. So thank you, everybody. And if you're here in the United States, good luck. Vote if you haven't already, and have a great day.
Thank you, speakers.
Thanks, everyone.
Thank you.
Thank you.